Hello, welcome to this week's VEuPathDB webinar on how to build complex search strategies using VEuPathDB databases. We will start just in a few minutes, giving a chance for people to sign in. And meanwhile, we would love to hear from you as far as what are the database or the databases that you use most frequently. I am showing the screen, I believe. Amar, could you let
You're
me?
you're not you're not yet, so you might have to click on share.
Okay. Okay.
I see it.
You
Oh, you
see
see
it?
it? Okay,
Okay.
good. Then
Excellent.
perfect.
So I'm showing a screen with a QR code to our live poll, and that also a link will be posted in the chat section. So go ahead and navigate and to the link and let us know which sites you use most frequently. I'll also will be showing the live poll on my screen, and there we are. And as you answer, we will be able to see this reflected on the screen. I will let the poll run for a few minutes and let others to join. And meanwhile, let me just mention a few housekeeping things. The webinar is being recorded, so if you miss anything, you will have a chance to review it later. Our webinars are posted on our YouTube channel, which is shown right here. And also, we will post the links on our site. If you're interested in receiving a certificate for the attending this webinar, it will go out in the email in a day or two, as well as with the link to the recorded webinar. Let's see, we have quite a packed outline for today. So if you have questions during the webinar, please go ahead and type them in the questions panel within the GoToWebinar panel. We are joined by several staff members of the VEuPathDB project, and they will be responding to your questions. And if some need a little bit further explanation, we will answer them during the webinar as well. So I see people responding to the survey, great. And it looks like people may be still joining. So if you just joined, please go ahead and fill in the survey, which is linked right here at the top or within the chat section of the GoToWebinar panel. All right, so I think we have a good number of people joined. So let's go ahead and begin. So my name is Evelina Basenko. I'm a FungiDB outreach specialist, and today I'm joined by several members of the VEuPathDB team. Suzanne Warfeld, located at the University of Georgia, and also Amar Harb, the director of the outreach and education of the VEuPathDB, located at the University of Pennsylvania. And today we will walk you through various sites of the VEuPathDB database and show you how to create searches using different types of data. If you're new to VEuPathDB resource, we have several databases available. I'm currently showing the main landing page for the VEuPathDB resource, and individual databases are also linked here at the bottom. The cool thing about our database is that they all have a similar structure or similar interface. So whether you are a researcher that using fungi or oomycetes in their research, you will find that the interface will look quite similar. So any searches that will be shown today can be recreated with different types of data for various organisms in different databases. If you would like to review our previous webinars are listed in the help section, which can be navigated from the help menu at the top. So today's webinar, for example, if you click on the explore button for the webinars, and notice that there's other types of information and help available. But if you navigate to the webinar page, we have upcoming webinars listed, and also previous webinars, where if you click on the link that you're interested in, you will have a direct link to the recording. And if there are any And if there are any additional handouts, they will be linked here as well. I would like to point out that next week our webinar will be concentrated on metabolic pathways, where you can learn how to find metabolic pathways and genes associated with pathways, and how also to paint not only experimental data integrated in VEuPathDB databases, but also phylogeny onto metabolic pathways. Okay, I'll go back to the main landing page and give you a brief introduction into navigation before I switch to Suzanne, who will show you the basics of the search strategies. So whether you're familiar with resources or not, I'll quickly go over with the main components of the landing page. We have a banner that presents a start search at the top that you can use gene ID or a text term to identify anything that has this records in the VEuPathDB sites. We also have a series of menu at the top that can help you navigate either to your strategies or specialized searches here, and also shown here on the left menu, various tools, workspaces, such as, for example, you can analyze your data in VEuPathDB Galaxy and then bring it back to the private spaces within the VEuPathDB databases. You can also look at the data currently integrated in our site, read more about the project or how to submit the data to us, or nominate datasets for integration. And we also have a help section that allows you to browse various types of resources, as I just mentioned before. And finally, if you have a question, you can always email us and we will try to respond to you within 24 to 48 hours. In addition, we have a section here on the right that has links to our social accounts. And notice that there's also login or register button. While you do not have to be signed in to use VEuPathDB resources, there are several services that require sign in. For example, if you are interested in analyzing your own data in Galaxy, you will have to register and sign in. And the data that we collect are very simple. We just need your email address to set up an account. You can also sign up for news to hear from us about various updates, new data sets integrated, etc. And we will not share your data with anyone else. So your information is safe with us. Also, at the top, underneath the banner, we have a section that's specifically dedicated to various banners. And here you can learn about upcoming events or some important things that we would like to announce. On the left, these are the specialized searches, they allow you to browse different types of data. For example, you can find genes that fall within certain phenotypes, etc. If you don't know where a certain search is located, you can also use the filter at the top. For example, if I'm looking for a signal, predicted signal peptides, I can search the filter at the top to identify the search that I want to use. And finally, the main page, the main sort of aspect of the page has overview of the resources and tools that are cards designed to get you started quickly with the website. For example, it can help you to to teach you how to initiate a search. If you're interested in looking at the transcriptomics data, you can learn how to deploy transcriptomics data search, or also view the tracks in JBrowse, etc. And a lot of these type of searches we will cover during today's presentation. And finally, there's also a quick link to the new section that you can expand or close off once you're done, that you can learn about the new data sets we integrated, or also the news and tweets that we post on our social media. I think this is it in just, so I will go ahead and transfer it to Suzanne, who will get you started on how to create search strategies in VEuPathDB resources.
Okay, you guys should see my screen. I'm open to PlasmoDB.
Yes, I can see it, Suzanne.
Okay. Woo, and I was worried there that the audio wasn't working for a second. Okay, all right, thanks. So I'm going to give a brief overview of our searches and strategies, how to build a strategy and the options for combining searches. Our searches are based on all of the data that we have. So let's review that. We have many types of data in our database, and the strategies allow you to query these types of data to assemble lists of genes with your desired biological characteristics. We integrate genome sequence and annotation, and then from that, we do a lot of genome analyses on that. We predict domains, we assemble orthology profiles using OrthoMCL, we add in gene ontology associations. We have a lot of expression data from transcript and proteomic profiling experiments. We have population genetic studies where we determine polymorphisms, single nucleotide polymorphisms and whatnot, a lot of clinical isolates, and then, just an array of data, right? Metabolomics, EST sequences, all kinds of stuff. So imagine if and not only do we have this data for just single genes, right? We have this for every gene in an organism and every organism in our database, and then all of the organisms that we support, which is now over 400 organisms. So, imagine you're a computer scientist and you have all of these tables of data at your fingertips. You could write a script that pulls out specific portions of the data. You could say, show me all genes that have 3 exons. Show me all genes that are expressed, during a certain life cycle stage. That's what our searches are. Scripts, and then we package those scripts into an interface that is hopefully friendly. And we've built ways for you to combine the results of subsequent searches to find groups of genes with shared biological characteristics. Even if you have 5 desired characteristics, you can do that. You can look for genes like that in our system. And we call that a strategy when you string searches together to find a group of genes that have the same properties or qualities. Okay, now I'll demo a basics strategy and show you how to run and combine these steps. I'll build a 2-step strategy that finds genes that are likely secreted. The data that we'll query are results of our predictions for all of these genes, whether they have transmembrane domains, and we also predicted whether they have signal peptides. And we'll combine those to queries to find genes with both of those features. So let's dive right in. I'll use this filter to find the transmembrane domain search. And I find it here under protein targeting. I click on that, takes me to the search page. I have a couple of parameters, right, that I need to fill out. Identify genes based on transmembrane domain. I have an organism parameter, and then 2 transmembrane domain parameters and a get answer button. The parameters are criteria that need to be met before a gene is returned by the search. So I want to go ahead and search all organisms for genes that have transmembrane domain counts between 0 and 99 genes, right? So I'm going to go ahead and click answer. And what I find, then I have some results to look at, right? I have a result page that shows me a growing strategy at the top, an organism filter on the side, and then a couple of tables on the left. And I went ahead and searched all organisms because this organism filter, what it returned to me is 75,000 genes, but I could use this organism filter to display only genes from, say, 3D7. Click apply. And now I'm not displaying 75,000 genes, I'm only displaying 1,730. And these are all transmembrane domain containing genes from P. falciparum 3D7 genome. So right now we have a single step strategy that returns genes with transmembrane domains. But what we said we wanted were genes with transmembrane domains and signal peptides. So I need to add a search that queries our signal peptide analysis results. So I'll click, and this is how I'm going to combine searches. I'll click Add Step, and I'm taken to this window, this pop-up, where I have a couple of choices. I can combine my transmembrane domain containing genes with other genes. I can transform them into related records. So I could say, show me all metabolic pathways that are associated with these genes. Or I could ask the question, say, show me other things on the genome that are near my transmembrane domain genes, and that's our co-location tool. But right now we want to combine our transmembrane genes with signal peptide genes. So I'm going to choose that option. And then it asks me how I want to combine these genes and what new list am I going to combine it with. So let's talk about a little bit about how to combine them. We have to choose that. So right now we have a list of transmembrane genes, and we're going to create a list of signal peptide genes. And just computationally, there's a couple of ways that we can combine these two lists, right? If we intersect them, we will find IDs that are in common between the two lists. If we union them, then we will have a non-redundant list of IDs from list 1 and 2. And we can subtract them too. We could say, show me genes that are unique in our first list, or show me genes that are unique in my second list. And really what we want to do is we want to find genes that have both of these biological properties, so they would be present in both of these lists, so we really want to intersect them. So I'm going to go back here and choose intersect and go to the signal peptide search and fill out that form. Select all organisms. It happens to be that we don't have to choose any other parameters, and click Run Step. And now I have a two-step strategy. My signal peptide search returned, about over 44,000 genes. The intersection of this is 720 genes. I've intersected that with my PF3D7 genes, which is why the number is so small. So really what I have done here, because this was PF3D7, I have pulled out the intersection of only the PF3D7 genes here. I can go back to this. Clear all of these so that my list now is the 75,000 genes that we started with from all organisms, intersect that with my list of signal peptide genes from all organisms, and I am left with 26,000 genes that have transmembrane domains and signal peptides. Okay, there's one other thing I want to point out, and that is, we don't just have searches that return genes. We have searches that return, single nucleotide polymorphism records or other types of records too. And that sort of, when we have two lists that are apples and oranges, one that's genes and one that's SNPs, the first four options of combining results are not valid, right? So when you combine these to intersect, you're asking the question is, show me all genes show me all transmembrane domain genes, show me all IDs in this list that are also in this list of SNPs. And there's just never going to be anything common between them. So we came up with this collocation tool, because the thing that we know about all of our records, all of our features, is the location on the genome. So we built this collocation tool to so that you can ask the question, show me all genes that have these single nucleotide polymorphisms near them. And you can define what near means. Show me genes that have single nucleotide polymorphisms within the first 100 base pairs, or in their latter UTR regions. So I think that is, you know, an overview of the strategy system. I wonder if there's any questions.
So I don't see any questions right now. I think one thing maybe you could point out, Suzanne, I'm sorry if I missed it, but the panel on the upper right-hand side of the strategy panel, you know, the save and the share, and what all of that
yes.
does.
I'm sorry. Yes. So, you know, once you have your results, you our database is a dynamic system, right? We're adding genomes, annotation changes, and stuff, and so you can save these strategies. You can add a description, so that you won't forget later on when you come back to look at this. You can actually save them, and what this does is save the parameters and the searches that you used, but not the IDs, because our database is a dynamic system, and when you run the search again, if you're in a future release with a different number of organisms or something like that, you'll get a different number of results. So when you save the search, you can go back and run it. You can also, once it's saved, you can share it with a friend. So we assign a unique URL to each of the searches. And you can copy that and send it to a friend. You can also delete that search, and won't be in your saved searches anymore. Right. If you want to download, so this will save the, you know, the strategy, how to set up the strategy, and rerun it on a new database if you open it up in a next release. But if you want to save the IDs, then you'll need to go to the gene results or the results table and use our download tool here. This will save the list of IDs that you've created with this strategy. Okay.
Cool. And one thing to note, that you notice that Suzanne is logged in, and you don't have to be logged in to use the database. So you can run this same exact strategy that Suzanne ran without logging in. But logging in allows you to save it in your history, and so if you want to make sure you don't have to replicate your work every time you visit the database, I would recommend that you log in and save your strategies. Even if you don't save them specifically with a name, they get stored in your history, and so you can always get back to them. And that's a nice feature available to you. Cool. So I think the next, yeah, switch it to Eve. Okay.
Mm-hmm.
All right. Thanks, guys. All right. I will demo a search strategy using VEuPathDB. And before we get into it, I just want to show you really quickly the steps, the operators, and the tools that we are going to use. So let's imagine for a second that I am a Candida auris researcher, and I am interested in learning more about genes that may be important for virulence. And what I would like to do is to use the datasets integrated in VEuPathDB for Candida auris, and then learn a little bit more about using data from other species, such as Candida albicans. So let's take a look at the type of data that we're going to use. We'll take a look at two organisms, Candida auris, and for Candida auris, we'll compare SNPs between geographically distinct groups of data collected from from clinical isolates. We'll also take a look at the genes that are related to these SNPs, look if any of these genes have predicted secretory signal peptide, examine the transcriptome data from the clinical isolates that were grown under biofilm-inducing conditions, and then learn from orthologs and Candida albicans and what is known for these genes when we take a look at the data set where Candida albicans were actually infecting mouse macrophages. And then we'll compare this data or intersect this data with what is known as far as the disease phenotype annotations in Phi-base. The operators that we are going to use, just as Suzanne have shown you, will take a look at the collocation, intersect data, and also transform data into other species. And this page will be available in the handouts if you would like to take another look. So let's go ahead and build strategy. This is the strategy, and let's navigate to the SNP data that we're going to use in our search. I clicked on the banner to navigate to the main page, and I'll go ahead and use the filter to identify SNP tool. In this particular case, I'm interested in using a search that looks for the differences between two groups of isolates. So I'll go ahead and click on this link, and we'll be redirected to a page where you can select from the drop-down menu the particular strain that you would like to work with. In this case, we will look at the data corresponding to Candida auris. I'm interested in a particular data set, and let me just increase the screen resolution a little bit here. The size. Here we go. We will take a look at the data set that is called Align Genome Reads Candida auris Clinical Isolates. And notice, because we're working with two groups of isolates, there are several portions for this page. For example, there's set A, which is our group 1, and set B, which is our group 2. So let's go ahead and first set up search criteria for the first group. We'll use this particular data set, and I'm interested in comparing the data from two geographical locations. First, for the comparison, I'll use data collected from Pakistan, and this data from whole genome sequencing of the clinical isolates from this country. And for the most part, I will leave the default parameters as they are, except for the percent of isolates. I would like to return SNPs only when 50% or more of isolates have that base call. And if you would like to learn more, by the way, about this search, or more specifically about the parameters, check out our previous webinars as we cover some of these searches quite extensively. So let's go ahead and set up the parameters for the second group, which is we'll select the same data set. And in this case, the geographical location that I would like to use is Colombia. So we'll be comparing two different countries. And I will change this to the 50% isolates with the same base call before the SNPs are returned, and click Get Answer. So notice that our page has now been refreshed, and I'll just limit to this. So the next step that we are going to do, we would like to identify, well, how many of these SNPs actually match to the genes in Candida auris. And I'll go ahead and click on the Add step, and we will use collocation tool. So what collocation tool does, as Suzanne already said, it compares essentially two different types of data. SNPs, which are individual positions within chromosome, with genes. And in this particular case, I will use the taxonomy tree to identify Candida auris as the organism of my choice. And I will use a filter at the top to quickly select the species that I'm interested in, and I will click on Continue. And while this, so the page has refreshed, and now the second step is to identify the search parameters. So we're interested to return genes in the second step right here that are overlapping with the SNPs returned in the first step. I'll go ahead and click Run Step, and here we go. Now we have over 5,000 genes that match our criteria, and they're listed at the table below. So the next step, I would like to learn how many of these genes has a predicted signal peptide. And again, I will combine this search now with another genes, and I will identify the search that I'm interested in running by using a filter and click on predicted signal peptide menu. Notice again, we presented with an option to select an organism, and I will click this search to identify Candida auris, and we'll click on run step, and I will intersect the step with the previous step as well. All right, so now we have 600 genes, and the next step that I would like to take a look at is actually look at the transcriptomics data available for Candida auris. I clicked on Add Step, and then I'll navigate to the transcriptomics section and click on the RNA-seq evidence. And as with other data sets, I will identify Candida auris transcriptomic set and click on the fold change search. So there are several things that we need to know about the particular samples. This data set is quite large, but basically we will be using Candida auris that have never been exposed to macrophages as our control, and then we will take a look at any genes that are upregulated in Candida albicans cells that are inside macrophages, such as live macrophages. So let's go ahead and do that. Oh, actually, I am a little bit ahead, so apologies. We'll take a look at this data set just in a little bit, but for now, I would like to go ahead and look for the upregulated genes that are induced under biofilm conditions. So I will use one of the clinical isolates grown under planktonic conditions as a control, and for the rest, I'll select different time points for the same clinical isolate that has been grown under biofilm conditions for 4, 12, and 24 hours, and I'm interested in upregulated genes. So let's go ahead and run the search and see what we return. So notice that how progressively the number of the gene changes, because we progressively sort of dig into what types of genes are either annotated with signal peptide predictions or expression in different types of data sets. So now that I have a list of genes induced under biofilm conditions in Candida auris, I would like to learn more information using other species and the data integrated in VEuPathDB for Candida albicans. So let's go ahead and transform our results, such as 221 genes, into Candida albicans. And I will click on the Add step to select the step that says Transform into Related Records. This is essentially a step that allows you to identify orthologs in other species to continue examine integrated data. So let's go ahead and click on orthologs and bring up albicans strains by using the filter search at the top. I will use Candida albicans at C5314 reference strain. And in this case, we will look for all orthologs, not necessarily those that are synthetic only. And I will go ahead and run step. So notice that now we have 366 genes, which is a little bit different from the 221 in Candida auris, and that's because some of the genes may have several orthologs hits. For example, this gene in Candida albicans have actually several orthologs in Candida auris. In the next step, what I would like to do, I would like to take advantage of the data that is integrated in VEuPathDB, such as transcriptomics data for Candida auris under conditions wherein Candida is infecting macrophages in mouse. And this is the data set that I brought up just a few minutes ago. So what we will use as a control is unexposed Candida cells, which means they were grown in media without macrophages. And we will use the comparison samples, such as Candida albican reads from the sample when the cells were actually inside the mouse macrophages. And we will do that by adding a step and selecting this data set from the transcriptomics data set menu, just as I shown before. So I will go ahead and I look for albicans. And the data set that I'm interested in is called Transcriptomic Analysis of Sorted Populations of Macrophages Infected with Candida albicans. And I'll go ahead and click on Fold Change Search. And this is something that you have seen before just a few moments ago. And I will look for the genes that are upregulated when Candida live cells are inside macrophages, mouse macrophages. So for our control samples, I will go ahead and select Candida only at all time points. And notice that there is your choices actually are described here on the right. So it's a graphical representation of how the choices that you're making for the reference and comparison sample are represented in your search, just to help you visualize your search conditions. And the next step, I will go ahead and select live cells. And actually, let's go ahead and select also those where the cells are dead, just to see, because some of the reads probably were discovered in the cells that may be dying. So let's go ahead and click those. But as you can imagine, because you have so many different time points in different samples, you can modify your search to fit your criteria. For example, you could look at the time frame only at 2 hours or only at 4 hours, etc. So let's go ahead and run this search. All right, so out of the 306 orthologs, 184 are actually upregulated during the time when Candida albicans are inside mouse macrophages. And there's several things that you can do with the type of analysis. As Suzanne showed you, you can look at the type of genes and investigate the information that we have integrated within the results table. You can also add columns if you want to bring up additional data within the table to view. You can download this data, or you can actually save this strategy in your account to come back and, let's say, build more or expand this strategy at a later time. There is, however, one more step that I would like to do. Notice that we have progressed from looking at the SNPs and ORAS intersected with the specific genes that carry the SNPs, then look if any of the proteins have signal peptides and how these genes were expressed in the clinical isolates grown under biofilm conditions, such as identifying only those that have at least twofold or higher expression. Then we look at orthologs and Candida albicans, and also use the transcriptomic data set that have looked at the transcriptomic response of Candida albicans during infection. And because we have various types of data integrated in FindGDB, including phenotype data, I would like to go ahead and take advantage of the manual curations that have been done for Candida albicans and add a step that specifically looks at the phenotypes. So let's go ahead and select the phenotype evidence. And from the drop-down menu, you will notice that there's a few data sets available. In this particular case, I'm interested in those that were integrated from the Firebase. And when I click on the curated phenotype button at the bottom, you'll have a lot of metadata that you can browse through to decide what type of data you would like to examine. But for me, I'll just run a search for anything that is connected to candidemia or candidiasis when it comes to phenotypes related to disease, and I will click on Run Step. And so essentially what I'm trying to do here, I'm trying to start by looking at Candida auris, learn what is known in Candida albicans, and then also learn, well, how many of these genes are actually annotated with virulence phenotypes. And out of 184 genes, I have identified that 9 of them actually are annotated in the Firebase curated data related to various types of virulence, such as 2 different types of diagnosis. So this is sort of in just how you can build strategies, and imagine that you can add multiple steps. Look for genes that also have SNPs using Candida albicans records, and much more. And I think I will stop here, and Amar will.
So you could, yeah. So maybe Eve, before you move on to Suzanne, for her, she was going to demo a strategy as well. I wanted to maybe make a couple of points here. I think what Eve was saying in the end, that really what you're doing is you're exploring the available data and resources within FungiDB in this case, and you're asking the question: can I find genes that are involved in infection? And she specifically was looking at genes that had particular types of SNPs and are potentially secreted. But the way you should look at this is you should look at it as an experiment, right? This is an in silico experiment you're running, and the list, the thing she did in the end, this intersection with the phi-base phenotype, basically confirmed that out of her 184 genes, 9 of them were already annotated by somebody else as potentially involved in infection. But the rest, the 184, right, you should look at this as if you're examining a result in the lab, and you will expect to find some background. You might find some false positives or false negatives. But as a scientist, you can look at this and you can then try and pull out and see if there are genes that belong to certain families or similar functions. Is this gene list, does it contain genes that you expect? And then if it does and you feel comfortable about it, then you can start exploring the other genes that are in that list, right? So if they're, just because phi-base, for example, found that 9 genes are involved in virulence, this is obviously not all of them, right? There are many more, and the 184 may include many of the other ones that you may be interested in exploring further and working on.
Yeah, that's correct. The records are constantly updated and the curated phenotypes are added, but these 184 are specifically essentially genes that are responding within when Candida albicans cells are growing within certain conditions, and in this case being engulfed by the macrophages, and they're either alive, dying, or dead.
And then the other thing to point out is that once you build a strategy like this one, you can, and you save it, you can always come back to it and decide, just like with an experiment where you may want to modify something in your experiment because you've maybe found out that if you increase the concentration of magnesium in your buffer, you get a better digest, right? So in this case, you could go back, for example, as Eve is showing, she's revising the step for the mouse macrophage infection. And she could, for example, change the fold change from 2 to make it 4, for example. She's looking for something that's even more differentially regulated, right? And so once she runs that, her gene list should go down in number, which is what is expected, but presumably a more stringent criterion that she's using.
Yep. And
cool.
Notice that there's some secreted genes that we have identified since we had a secreted peptide chosen, but imagine that you may want to delete these steps. You may not be interested in no longer having this step, so you can always go ahead and delete the strategy. And to keep you, if you save this strategy, it's we have a neat tool where you can change the names for individual steps, and that's also a good idea to do if you want to sort of remember what is it that you have searched, as well as you can create the description within each strategy to remind you of the type of search that you ran if you want to save it for later.
And maybe one last thing to demo before we switch it over to Suzanne is, as we indicated, you can share these strategies with a link, but you can also make them public. And so maybe what you can do is, Eve, you can demo making this strategy a public strategy, then everybody on the call can go to the public strategy section and they can open this strategy if they want.
Okay, it is being saved as a name strategy, but I will go ahead and save it under today's webinar date.
Cool. And I guess we don't have to necessarily show that, but you can do that. But then people, if they go to FungiDB, you can go to the public strategy section in the My Strategies panel, and once you're there, you will see the same exact strategy that Eve is currently saving. And she can always go back and update this later on.
Yep.
But this will show up in the public strategy panel. Mm-hmm.
Yes, I'll make it public. For publics, you have to have a little bit of a description in order to post it, but here
right.
we are,
And you can
so
do
it's
that.
accessible.
Yep, perfect. Thank you, Eve. All right, so we should switch over to Suzanne now. And let me see if there are any questions. So again, if you have any questions about how to build a strategy, let us know in the question panel, and we'll go ahead and answer them. There aren't any right now, so Suzanne, it's all yours.
Okay. Y'all can see my screen?
Yes, yes.
So I'm going to build a strategy looking for possible druggable targets for bradyzoites in ToxoDB. So for Toxoplasma, the definitive host is the cat, but it also invades other species, including human, where it's present in two stages: the tachyzoite, which is the invasive and rapidly dividing form of the acute infection, and the bradyzoite, which is a latent form that's encysted by the host and is present in a chronic infection. In the cat, it's present as a merozoite, and it forms oocysts, which are secreted, and that's what infects us. So if I wanted to find a protein to target in bradyzoites to develop a vaccine or a drug, I would want the protein to have certain characteristics, right? And here's a couple I could think of. I would want it to be upregulated in the chronic phase, right? Upregulated in bradyzoites. It would nice if it would be not highly expressed at the merozoite or tachyzoite stages. Be great if it didn't have any orthologs in humans, right? And it would also be great if we could show that there was evidence of protein expression in the bradyzoites. And then, depending on what type of drug you wanted to develop, you might look for genes that have a specific enzymatic activity. So those are the stipulations I'm gonna put on my search, and I'm gonna go ahead and start a search. And it turns out that we have a data set named Transcriptome during acute and chronic infections in a mouse brain. It was done by the Laura Knoll lab. And what they did was they had compared uninfected, acute, and chronically infected mice that were infected with T. gondii. They looked at the forebrains and collected RNA from that, and then did RNA sequencing. So we know the gene expression at the acute phase and at the chronic phase. So I'm going to go to ToxoDB and look for that search. On the RNA-seq search page, I'll type in chronic. And here we are at the search. I'm going to use our differential expression based on statistical analysis. And that is asking me to define several different parameters here on the left. I will look for, I will compare the chronically infected mice at 28 days to the acute infection, look for genes that are upregulated at a fold difference of twofold with a p-value of 0.01, and I click Get Answer. Whoa, and I'm already down to about 200 genes, right? Exactly 200 genes. So these are genes that are upregulated in the chronic phase versus the acute phase of bradyzoites. Now, we said we wanted to possibly avoid genes that are highly expressed at other stages, and so I want to subtract from this list another set of genes that are, highly expressed or, in the upper expressors of merozoites and tachyzoites. And I've looked through our data, and I have found an RNA-seq experiment that looked at tachyzoite and merozoite expressions. So I'm going to combine my upregulated genes with another list of genes that I want to subtract my new list that I'm going to create from an RNA sequencing search. And the experiment that we're going to query is the tachyzoite and merozoite transcriptome from the Heil lab. I'll use the percentile search. And this search allows us to return genes that are expressed at based on the distribution of all gene expression within that sample. Okay. So for this experiment, I will say, yes, I want to query the tachyzoite and merozoite samples, and I want to return genes or subtract genes that are highly expressed in the 80th to 100th percentile of expression values within these two samples. And I'll click Run Step. And now I have a two-step strategy. My percentile search returned 2,300 genes that were in the 80th to 100th percentile. The intersection of these genes with my upregulated and bradyzoite genes is only 74 genes, right? So we have 74 genes that are upregulated in bradyzoites and not well expressed in the other stages. Now, the next thing we said next quality was that we wanted to exclude from this list genes that have no orthologs in humans, right? So I'm going to add step and intersect that with an orthology phylogenetic search. This is a really unique search that is very, very useful. So when we incorporate all of our genomes, we run OrthoMCL and assign all of our genes to orthology groups. And from that, so we have this array of clusters of orthologs, right? And from that, we can, because we know what genes are assigned to each of those clusters, each of those groups, we can create a profile for a certain set of genes. And then we developed this search, the orthology phylogenetic profile search, that allows us to search for, exclude genes that have orthologs in certain groups. So I'm going to choose select all in the organisms, and I want to exclude genes that have orthologs in mammals. Okay. So I put a little red X there. So the genes that are returned by this search can have orthologs in any of these other species, but not in mammals. I'll click Run Step. And it returns to me 164,000 genes that do not have orthologs in mammals, and my intersection with these genes is 56 genes. So we've been able to exclude another 20 or so genes. And these genes are upregulated in bradyzoites, don't have super high expression in other life stages, and do not have orthologs in mammals. Our next property was we thought it'd be great if these genes had protein expression. So we will intersect that with a proteomics search. And the data that we're querying here is proteomics data. We have integrated experiments where people have isolated protein, done proteomics, and we have mapped the peptides that they found within samples back to the genome and correlated that with genes. So we're looking for genes that have mapped peptides in these proteomics experiments. And I would bet money that we have an experiment that was done in bradyzoite right here, mouse brain bradyzoite proteomics time course. I'm just going to go ahead and pick all of those samples and click Run Step, now we're querying that proteomics experiment for genes that had mapped peptides in any of those samples that we chose. And we returned 1,300 genes, and the intersection of those genes with our last list is 22 genes. So these genes are upregulated in bradyzoites, not highly expressed in the other life cycle stages. They do not have orthologs in mammals, and they are shown to have protein expression to be expressed at the protein level during from other bradyzoites. And then, we can go on and on. We could look for other enzymatic activity, possibly just from a text search. Look for kinase genes. But the interesting thing to do is to look through our genes, right? So now we're down to 4 genes. I'd say any of these lists are great to, possibly look for more information about these genes and then take them back to the lab to start investigating. And the great thing here, because you're doing an in silico experiment, you're finding things that possibly are not already correlated with with druggable targets. So here, our list of 4 genes has 3 hypothetical genes and 1 gene that's a alpha-glucan dikinase gene. So that's my strategy. I wonder, is there something else you were thinking of, Omar?
No, I think this looks great. I mean, I think the point here, we are there are many ways of searching these strategies and different ways to integrate them. And so the question, I guess, that's coming up, and I see it now in the panel, is, for example, if you had you were going and exploring the data sets that are available, but is there a different way where you can find what data sets are available in for example, in ToxoDB? Where can you see all the data sets that are available and maybe search them there?
Mm-hmm. Yeah, we do have a data sets page that is totally searchable from the upper header, the data menu, data sets in ToxoDB, and this is the same on all of our sites. You can go to that. We've applied the same ontology that we do that we use on all of our sites to the data sets, so they're categorized, and those lists will correspond with searches and stuff like that. So if I wanted to browse, I could say chronic, and there are 3 data sets, 2 RNA-seq and 1 microarray that deal with chronic infections. Another example would be bradyzoites. Now I'm showing 13 data sets out of 157 in ToxoDB that reference bradyzoite. So there's quite a few of them, actually. A lot of them are transcriptomics, a bunch of DNA microarray expression, some 2 protein expression, and 4 RNA-seq. There's actually also a SNP data set that has to, that will reference bradyzoites.
Cool. And
yeah.
Yeah, no, that's great. I think that's exactly what the question was. Trying to see if there are any other questions. I guess one point to make is that if you are working on one of our databases and you notice that there's a data set that is missing, you think, oh wow, this data set was published recently or whenever, and it seems like it's really good data to be available for the entire community within the database. Definitely contact us. So you can click on the contact us link. We also have a data set submission page where you can propose data sets. But if you're having the easiest way often is just to contact us, and we can start the conversation there, and we can point you in the right direction if needed. Eve, I think we're at the top of the hour. I'll let you maybe finish up with the survey.
Yeah, sounds good. Thanks, Omar. Let me go ahead and share my screen. I just posted also a link in the chat section, and that is a link to the webinar survey. So we would love to hear what you think about today's survey, how we did, and today's webinar. And there's also an option to suggest topics for future webinars. So do click on the link, fill it out right now if you can, because we know that once you navigate away from the webinar, it's quite difficult to come back. It's a very short survey, but it helps us tremendously to learn about your interests, how we can improve, and how we can make the dedicated webinars of better use to you as a researcher who uses our databases. So I think if there are no other questions, we probably will go ahead and finish up. So I would like to thank you all for joining. And as always, if you have questions regarding anything that has been covered today or just in general, click on the Contact Us link in any of our resources. All right, thanks very much. Bye-bye.
Bye.
Bye.